Okay, I think we should go ahead and get started. I'd like to introduce you the speaker for today's ERI seminar, Professor Steve Lindo. For many microbiologists, Steve really requires no introduction. I may embarrass him by describing him as the granddaddy of plant microbiome research, phytosphere microbiome research. But he really is. He did a lot of foundational work that influenced our understanding of how bacterial life adapt to and interact with plants through the surface to this day. So Steve has been a faculty member at UC Berkeley for a long time, and he's also a member of the National Academy of Sciences. I think that's sufficient credential for you to take him very, very seriously. Thank you.
Thanks, Jerry. Thanks for coming to listen. I'm going to try to cover kind of a broad perspective on the very issues that relate to microbial life on leaves. I know many of you would have heard kind of microbiome research. Certainly gut microbiomes in humans and animals is big these days. And even if you've heard about plant microbiome, it's usually been on the roots. But today we're going to talk about the microbiology of the aerial parts of plants. Because we think it's important and try to convince you why.
That's the fact that there's the
Focus on Zoom.
no. It's the you have to turn off the laser pointer. Maybe.
Okay. The foliar microbiome is probably almost as large as the microbiome on roots itself, in that you see here a stomata for size comparison, and all these little objects all across the surface here are all bacterial cells, mostly bacteria and a few fungi. Plants are all colonized with very high numbers of microorganisms, and they vary from about 10,000 to about 100 million bacteria per square centimeter. When you add them up over all of the leaves and above ground parts of the plants in the world, you get about Avogadro's number of bacteria, probably larger than that of the ocean. But it's quite a large number, and they're very important for various processes. And we'll talk about that today. And much of the studies of plant microbiome has been coming from the study of plant pathogens historically, because we know that many plant pathogenic bacteria live as healthy microbes on plant surfaces. So we think of plant pathogens such as that causing bacterial brown spot of bean and fire blight disease of apple. These exist for most of their life as a saprophyte on the surface of healthy plants. And previous picture, we showed there's various large numbers of bacteria. Most of them are not pathogens, but they have a variety of other traits that we're going to talk about. So most of the work on foliar microbiology come from the study of bacterial plant pathogens. And it was possible to predict disease based on the population sizes of these bacteria as basically reservoirs of potential pathogens living on the surface of plants. As bacterial numbers increased, the probability of disease would also then increase. But we also know, though, that most of the bacteria on plants are not plant pathogens, and they have many other traits that we want to be talking about. And most of those are, in fact, protective of plants. And most of these non-pathogenic bacteria actually provide considerable protection against plant pathogens, because when applied preemptively, they can prevent the growth of pathogens. And a classic example of that would be that of fire blight disease of pears and apples, where the flower parts of plants emerge into the world nearly sterile. And this is something we're going to come back to in several different ways. But the female part of the flower is basically sterile when it emerges, and then if the pathogen is then brought to that open habitat by an insect, in this case carrying the pathogen, Erwinia malavera, that pathogen can then grow to high numbers, and disease is then very, very likely to happen. But we can know, though, that if various non-pathogens arrive in that habitat first, that largely prevents the growth of the pathogen, which you see the little red jobs, that those pathogen populations remain quite low and disease can be prevented. So just a simple example then of how many of the microbes on plants are in fact protective, just like gut microbiomes are protective against human pathogens. But it has a practical implication, because In fact, there are a variety of non-pathogenic bacteria, including Pseudomonas fluorescens strain that we found some years ago, that when applied preemptively onto plants, in this case, almond trees, can be can prevent then the growth of the wrong bacteria, if you would. And one of those wrong bacteria that we're going to talk a little bit about is that of ice-nucleating bacteria. That is, we were all taught incorrectly that water freezes at 0 degrees Celsius, but that is not true. That water has the potential to supercool, to remain in a liquid state to quite cold temperatures in the absence of a catalyst for that liquid to solid phase transition. And certain bacteria that are quite common on plants, like Pseudomonas syringae, are very efficient as acting as a template for catalyzing ice formation. We call ice nuclei. In this case, they can catalyze ice when water cools only to around -1 or -2 degrees Celsius. In the absence of these bacteria, the water will easily supercool down to -10, or in small droplets, pure water, to around -40 degrees before ice will then form. And this is important then because plants, many of our agricultural plants are very susceptible to freezing. And it's not the cold temperatures that causes frost damage, we call it, but in fact, the formation of ice in the tissues. And it's the presence of these bacteria on the plants that then elicit then the frost damage. So much of my work then has been driven by an understanding of the microbial ecology of plants, specifically related to ice-nucleating bacteria, to avoid or to enhance the supercooling ability of plants. So in this case, you see that plants that were grown in a sterile condition, corn and beans and so on, without the bacteria present, can easily supercool to around -6 degrees. Ice nucleation is very, very unlikely at temperatures above that temperature in the absence of these bacteria. And in fact, then the freezing temperature of the plant is driven then by the populations of these bacteria. So if we have high numbers of bacteria, like 1 million cells per gram of tissue, the plants will supercool no more than to around -2 degrees. But as we decrease the population size of bacteria, we see then that the temperature at which the plants can supercool to before they're likely to freeze approaches that temperature of around -6, where they're going to freeze no matter what. So plant ice formation in plants then is largely driven by the presence of these bacteria. And if you were foolish enough to get up in the middle of the night and watch plants freeze like I've done here, these citrus leaves in the upper left show that darkened area near the center of that top leaf is where a bacterium has triggered ice formation, and ice was slowly spreading through and across that leaf surface. And that is what happens thousands and thousands of times in a plant canopy. But again, it seems to all be driven by the presence of these nucleating bacteria. And just like the presence of non-pathogenic bacteria can preemptively prevent the growth of pathogens, non-ice forming bacteria can also prevent the growth of the ice-nucleating bacteria. So in this case, these plants were all put in a freezer at -5 without any bacteria grown in the greenhouse, reasonably devoid of bacteria. They obviously supercooled and were not damaged even at -5. When we inoculated only with a nucleating bacteria, they grew to high numbers and it was doomed to freeze at a temperature around -2 or so. But in the middle, you see that those plants were first inoculated with a non-nucleator, and then the nucleating bacteria then could not grow to high numbers, and you see most of the leaves escaped any damaging ice. Now, the ice nucleation phenomenon we'll talk a little bit more about is really interesting in that it's encoded by a single gene in the bacterium. These are very large genes for bacteria. They're in 4 to 5 kb in size, and they encode a very highly repetitive and encode a repeating 48 amino acid sequence, randomly repeated over and over and over, some 20 to 50 times. So they're very large proteins, which this basic unit, this 48 amino acid block, computer modeling shows to form these little rectangular structures that are on the surface of the membrane, which have hydrogen bonding groups from serine and threonine, which overlap almost exactly with that of an ice crystal you see on the right. So if you look at this, the hydrogen bonding oxygen molecules, for example, on the ice crystal, it matches exactly their hydrogen bonding pairs in the ice protein. So it basically acts as a template upon which water is held into a structure that looks like a ice crystal. and then would allow it to grow spontaneously. So it's an interesting biological phenomenon, but it has some really important applications. So here you can see artificial snow production. If you've ever gone skiing, most ski resorts make artificial snow anymore, and most ski resorts that make artificial snow use the bacterium that I discovered to catalyze ice formation. You can buy it, it's called Snowmax. It's a freeze-dried, gamma-irradiated culture of Pseudomonas syringae, mixed with the water, sprayed up in the air, and the ice freezes in the air as ice crystals rather than falling as freezing rain, if you would, to the ground. So it became a big project for which I get $0, but the world is a better place, I guess, because we can make artificial snow. But it's also very important in the greater scheme of things. In the global precipitation process, there's thought that this is one of the major players in rain and snow production naturally. So we'll be today talking a lot about the growth of these bacteria on leaves, and a little later on, I'm going to talk about their escape from leaves to go up into the sky. Quite large numbers, about 1 million per square meter per hour, escape and go up into the sky. And they would interact with clouds. The clouds we see are almost all below freezing. They're all super cooled water drops. They're basically cold fog. And that fog, just like it doesn't rain out of fog, it needs something to make those drops big enough to fall out of the sky. And that requires ice formation in the clouds. And so we see then a bacterial cell interact with one of these super cool drops. It will cause that to freeze, and by a couple of different processes, make those particles of ice get bigger and bigger in the clouds, and eventually they fall from the clouds because they're too large to be held up there. If it's cold all the way to the ground, we call it snow, but most of the time it melts on the way down and we call it rain. But most of the rain in the temperate world, except for very close to the equator, starts as ice. So we've had recently rain here. Those were all triggered by ice formation, and a large percentage of that is thought to be these bacterial cells because they're active at such warm temperatures. Meteor dust and certain minerals can also catalyze ice, but much colder temperatures. -10 to -15 degrees. These bacteria catalyze ice at -2. So even warm clouds can cause the formation of these precipitation. So it's thought to be quite important in the global, like I say, the water cycle. And that's led people to speculate about, again, one of the many ways that people are screwing up the world, if you would. In this case, the Sahel region in northern Africa is increasingly drier and drier over the decades. And the Sahel region used to be kind of green. There used to be grasses and all, but humans brought in goats and sheep and removed much of the vegetation. And that vegetation removal, there's a lot of speculation, is allowing that or preventing the development of ice-nucleating particles to get into the air in that part of the world. Can't prove it. It'd be a very difficult thing to prove, but it's thought that it might be interrupting the global precipitation cycle because of that. And if you ever look out of a window, certainly if you're flying across the United States, the western part of the states are pretty arid, but towards the east it can more and more wet. Much of the central North America, including most of the United States, used to be a large grass-covered prairie. It's now been cultivated, and now we grow corn and soybeans, as endless expanses of corn and soybeans. In the spring and early summer, it used to look like upper right. It was covered with green grass, and you can imagine plumes of these bacteria making their way into the sky. That clearly isn't happening anymore because it's been denuded. We see the cultivated land for much of the year. There'd be many parts of the world, many parts of the year where there wouldn't be a source of these bacteria. So again, is this precipitation process going to be altered by agriculture in many ways? Again, hard to prove, but it's something that people worry about. And again, it might be an effect on climate. And we talk about climate and CO2, but there may be a direct effect of these bacteria on the climate because of the change in the radiation balance of the Earth. So we see light coming from the sun. Much of it is reflected back into the sky by clouds. And the role of these ice-nucleating bacteria, ice-nucleating particles in general, is to make the clouds brighter. Clouds that have large numbers of small particles are brighter. They're more reflective of the sunlight than clouds that have bigger droplets. And it also affects the way in which the radiation can escape back into space by making the high clouds, the very high cirrus clouds, thinner. So it might be a very direct effect by making the clouds darker than they normally would be by reducing the numbers of these airborne particles. So again, the cloud physicists will have a lot of interest in what we're doing on plants because of this effect, direct effect on the radiation balance of the Earth. So let's get back to Earth then on the leaves where this is all happening. What is driving these populations? It seems to mostly be the amount of potential carbon to be consumed as food by these microbes. And this is basically on the menu of a plant. So the primary sugars, glucose, fructose, are the major carbon sources on most plants. There are a variety of other lesser molecules, certain organic acids and amino acids. But note the units, they're micrograms or so per leaf. It doesn't sound like very much, but if you do the stoichiometry, you can generate around 10 to 100 million bacteria with the 10 or so micrograms of carbon that would be on a typical leaf. And it turns out that plant species vary in how much of these nutrients that they have. Certain plants like tomatoes and beans and all have relatively high amounts of carbon available, and other things like peas and corn and so on are much less. And it seems to be largely structured by the cuticular surface. The diffusion of nutrients from inside the plant is restricted on many plants, and so there's much less available carbon to be eaten. But the important point I want to point out is that the nutrients that are available for consumption are not uniformly distributed. So we made a variety of these biological sensors, and the first one I want to talk about is a carbon sensor for fructose in this case. We take a fructose-dependent promoter and have it drive an unstable variant of GFP. So the cells harboring this synthetic gene will only produce green fluorescent protein if fructose is around. And they will, because the protein turns over quickly, it will remain green only if they see fructose at the instant. And when we grow up a biosensor like that, grow it up without fructose around, so there's not fluorescent, spray it on a plant, come back an hour or two later, you'll see that there are little green cells all over the surface of the plant. But if you come back a day later, there's still cells all over the plant, but there are only a few areas where the nutrients are apparently still available to allow the production of the green fluorescent protein, which we interpret then as being just a small wisp of nutrients over most of the plant, and only a few areas where nutrients are actually relatively abundant and might continue to come to the plant. If we think of this as kind of a nutritional oases, they're relatively uncommon on the plant, but it also seems to be where most of the action on a plant comes from. Because if you do an imaging, like one of my students who took thousands of images of the plant surface, you can see this kind of sparse colonization of the bacteria over most of this image. And then you see this large object in the middle where we see a microcolony. There might be 100,000 or so bacteria in this colony. Most of the rest of the part of that image is largely devoid of the bacteria. And we think this is driven by the fact that these bacteria are being fed by that local oases of nutrients where they're coming out. Often at the base of trichomes and then other places where we can't really be sure why there are nutrients there, but presumably it was driving the potential to grow large colonies of these bacteria. And these oases are relatively uncommon. If you look at large numbers of these images and look at the number of aggregates that we see on a plant as a function of how big they are, you see we have literally thousands, and the y-axis here on the top is on a log scale. You have thousands of times where you'll see a few cells together in a given location. And as the aggregate size gets bigger and bigger, you see there's only a few aggregates that are so large. So we get an aggregate of 10 or 100,000 bacteria. There may only be a couple on a leaf. But if you look at the bottom graph where we're looking at the cumulative proportion of all the bacteria that are on that leaf as a function, again, of aggregate size here on the x-axis, and let's just use 100 cells as a cutoff, you'll see that about half of all the bacteria on a leaf are in these relatively large aggregates of 100 cells or bigger. And that these individual cells that we see all over the leaf don't add up to very many of the total population. And that reminds me an awful lot of human colonization. Here's a demographic map of the United States. Any country is going to look similar. So, we live over here in Berkeley, and if you were to fly from there over to New York, you're going to fly over a lot of farms and desert where there's hardly anything when looking down on the planet. There's very few people there. But then you eventually get to Chicago and you get a huge city all of a sudden. California is the most urbanized state in the United States. If you've ever visited, we got huge deserts and forests and mountains. But most Californians live in cities, and that seems to be the case with the microbes. There are reasons why we live in the city because there's jobs there and water systems and all that have been engineered to allow us to live in these places. And I'm going to talk about microbial engineering of the leaf in just a second, because the microbes live in these few places where they do live because there were already resources there, where I'll talk about how they actually are modifying the leaf to live where they do live. But before we do that, let's think again about these microbial cities. It seemed to be very important to be in a microbial city. Not only is that going to be a good place to grow up, but it's also a good place to stay alive. And so what we're seeing here is the fraction of living cells as a function of the size of aggregate that they're found in. Let's just use 100 cells as a cutoff again. You see all those cells that were in these relatively small aggregates, 1, 10, 50 cells, they're mostly dead after they've been exposed here at the yellow line for wet and dry conditions for 7 days. And we think the drying is particularly detrimental to the survival of the bacteria on the leaf, and they're not surviving when they're part of these individual cells, very small individual cells. It's only when they become part of these large aggregates are they actually alive. So these images that you saw earlier of the bacteria spread around the leaf, most of them were probably dead under the kind of typical environmental conditions that they have to survive out in the real world. But it also has other practical implications about how does a leaf become colonized. And we'll talk about the immigration of inoculum to the leaf here in a little bit. But it turns out that especially if you're not a particularly fit organism for growing on a leaf, like certain human pathogens like salmonella that we don't want colonizing our tissues, they can in fact survive quite well if they land amongst one of these large aggregates on the leaf. So in this case, we had some differentially color marked strains of salmonella, and when it landed then on a empty part of the leaf without any other bacteria around. And the y-axis here is again the percentage of the cells of salmonella that were actually dead after 24 hours after we'd inoculated. And you see, contrast that with the dark green bar where it landed amongst some other cells. It was largely alive. It landed in this big pile of other microbes, and that seemed to allow it to then survive. So the successful immigration of a microbe to a leaf is also driven by the pre-existing communities. The microbes that were already there are protective. So this then seemed to be a better image of what a life on a leaf looks like. So here's one of those aggregates. This is a scanning electron micrograph. And you see it's kind of covered with this polysaccharide or slime that you can easily imagine being quite protective. On a sunny day, the sun is coming down, that slime is probably acting as somewhat of a sunscreen, it's probably binding water. That is a group that was growing there because of the resources allowed to develop such a large population and to produce enough of that protective slime. So again, that is basically what we think is where most of the microbes on a leaf are actually at. But a microbiologist would look at that and they might also say to themselves, quorum sensing, that they may be density dependent behaviors, which has become quite a hot topic in microbiology, and that we know that microbes, although they're individual cells, have a programmed changes in the way they behave when they are part of a group than when they're more solitary. That's what we call quorum sensing. In this case, it's driven by their ability to produce and perceive these small signal molecules. One of the most common ones and the most studied is that of these N-acyl homoserine lactones that are produced at very small amounts constitutively by the bacteria. When they're alone in the world, that molecule can diffuse away from the producing cell, as you see up here in the upper left. And it doesn't see the signal molecule anymore because it's diffused away. So it knows it's solitary, and so it has a programmed behavior that's appropriate for that kind of setting. But when it becomes part of a large group and all of its neighbors are also producing the same signal molecule, then it changes in its regulation of different traits. And human and animal pathogens, many regulate their production of virulence traits by the reception of these signal molecules. Pseudomonas syringae is opposite. It actually suppresses many of its virulence traits when it's part of one of these large groups. In this case, Pseudomonas syringae, a very common leaf colonizer, when we make mutants that are blocked in its ability to produce and perceive the signal, it actually becomes more virulent, and probably because it also is more motile. Here, a wild type strain did not want to leave that colony, but when it was unable to perceive the signal molecule, you see it's now swimming away from where the colony was originally inoculated. So the signaling process seemed to be very prominent in many species like Pseudomonas, but it also then raises this question about whether the behavior of a microbe is really under its own control. How much of it is going to be kind of happenstance or controlled by things outside of its own control? In this case, these bacteria have other neighbors on a plant. And those other neighbors, we now know, can produce the same signal molecule as, in this case, the plant pathogen, Pseudomonas syringae. We found that about 12% of the bacteria on a plant make a signal molecule that Pseudomonas syringae, the plant pathogen, can also perceive. And interestingly, we find about 8% of them can produce chemicals that interfere with the signaling of the pathogen to its own signal molecule. So again, we think of it earlier as Pseudomonas thinks it wants to become virulent, so it produces or doesn't produce a signal molecule. But what if it has neighbors around which has other ideas? Can they then influence the behavior of Pseudomonas? And obviously the answer is yes, or I would be talking about it. In this case, we have neighbors that produce the same signal molecule as Pseudomonas syringae. In this case, it's a strain of Erwinia that makes a large amount of the signal molecule, maybe 100 times more than the pathogen itself normally would make. When we inoculated Pseudomonas syringae alone on the left-hand bar, and we let it grow on the plant for a couple of days and look at how many lesions that it made in this case 35 lesions. When we had mixed it with Erwinia and inoculated the plant, you see the number of lesions went way down. But it might have been due to competition and other things that were responsible. So to account for whether it was simply competition versus this communication change, we blocked the ability of the Erwinia to produce a signal by adding a gene enabling the production of an enzyme that simply destroyed the signal. And there you see then that the amount of disease is very similar to that of the pathogen by itself. That it was the presence of this extra signal molecule which stopped Pseudomonas syringae from moving around on a leaf, prevented its ability to get into the leaf where it could cause disease, that was responsible for disease. And the opposite was also true, that when we had strains like this strain 114, which interfered with Pseudomonas syringae's ability to make signal, now compared to the pathogen alone, we see like 2 or 3 times more likelihood of disease because the cells are more active. They are not suppressed by the presence of the signal. They're moving around and apparently finding their way into the plant where they would then cause disease. So again, disease is very conceptual. Now, what are the traits that are needed for living on a leaf? The bacteria that live on plants seem to be rather unique. Many soil organisms do not survive at all well on a leaf. And so we were very interested in what are the traits that seem to be particularly important among these bacteria that grow on the surface of plants. So we were fortunate that one of the earliest strains sequenced was Pseudomonas syringae by the Joint Genome Institute. And so we had in hand then the ability to start doing transcriptomics of this bacterium early on. And we were very interested in what are the traits that were expressed when it was growing in culture compared to when it was grown on the plant. So we have now basically a histogram of the patterns of gene expression that we saw when the bacteria were on the surface, when they were on the inside of the plant, or when we grew them in a middle medium that had salt and peroxide and low iron and various conditions that we thought might mimic what the leaf surface looked like. If you just look at this tree showing the basic patterns of gene expression, not too surprisingly, we see there's a different pattern of the genes when the bacteria we removed from the plant before doing transcriptomic as compared to on the inside of the plant. But quite concerningly, you see that the pattern of gene expression that we saw in any of these other culture media were very, very different than the patterns we saw when they were in or on a plant. So for a long, long time, people have been studying these bacteria in culture. But in fact, they had very, very different behaviors when they're, in the plant, in or on the plant. Probably something we should have guessed, but it's very disconcerting that in fact, a lot of what we learned about in culture probably wasn't telling us very much about what was actually happening in and on the plant. But just a couple of snapshots about what are the kind of traits that seem to be particularly highly expressed when we were on the surface of the plant. We'd washed the cells off and done transcriptomics. One of the things that really stuck out was that all the genes that were involved in flagellar motility and chemotaxis were upregulated, you see in the red bars. And another trait that we're going to talk a little bit more about later on would be tryptophan biosynthesis. It wanted to make a lot of tryptophan when it was on a leaf, and we'll come back to that in a minute. And conversely, when you got the bacterial cells out of the inside of the plant and done the transcriptomics, what are the traits that seem to be particularly important? There were a lot of them involved in plant toxins, things that would damage plants. And this is something we knew already quite a bit about, but both cells on the surface in the red, but especially inside the plant in the green, these several different toxins were all induced. Okay, but what was really quite interesting was this other group of these compatible solutes. These are molecules like NEGGN, proline, trehalose. These are all molecules to allow the cells to tolerate low water conditions. And we would expect them to be upregulated on the cells that were on the surface of the plant because that dries up frequently. And you see that in the red that yes, cells off the leaf surface were trying to survive desiccation conditions. But look at the green bars, which are the cells that were on the inside of the plant. They were even more strongly upregulated, suggesting that the inside of the plant that we would have all considered to be kind of moist and juicy was actually very dry. And it is, because you think about how plants work, gases have to diffuse into the intercellular spaces to supply the carbon dioxide for photosynthesis, and also the cells lining the inside of a plant are actually mostly separated by air. And the cells that were on the plant or inside the plant had to tolerate that very dry condition. And one other trait that we're going to talk a little bit about in a second is syringofactant, one of the many biosurfactants that are produced by the bacteria. They were upregulated both in and on the plant. So yes, there were very different traits that were upregulated when you were on the inside of the plant and when you were on the surface. That's what we might expect. But the traits that I want to follow up on would be a couple of these related to syringofactant production with these biosurfactants, and also tryptophan. Why was it trying to make so much tryptophan when it was on the leaf surface? Let's talk about tryptophan first. Tryptophan is the immediate precursor for one of the most important plant hormones, indole acid. So if you think back to your biology, auxins, indole acid is one of the major plant hormones that have evolved in plant development. In plants as well as in bacteria, these are all ultimately derived from tryptophan as the immediate precursor. It turns out that virtually all bacteria that you see on a plant produce indole acid. Very few don't make indole acid. And it seemed to be very important for them to have made indole acid. And it also suggests that many of the traits that we see, the normal traits of plants like flower abscission and fruit abscission, or the development of epidermal cells here to make what we call fruit russet, is regulated and produced because of the auxin that these bacteria are making. So the fruit russet is an important agricultural trait where the cells of the fruit die prematurely as the fruit matures. is very big economic problem, especially in California, where we grows a lot of fruit for fresh market compared to a fruit where we did not have the auxin producing bacteria present. It's again thought to be something that was originally driven by the plant and weather, but it's actually driven by the microbes that are living on the plant that are making the hormone. And again, it seemed to be very important for those bacteria that make the hormone, because when we made mutants of the bacteria that were blocked in that pathway that converts tryptophan to indole acid, and we applied an equal mixture of the wild type and an auxin deficient strain onto the plant, and looked at how that ratio changed after we inoculated on the plant, you see that basically there was a substantial increase in the ratio of the auxin producing to the non-producing strain, suggesting that the auxin really was important, even under short periods of time, for its ability to do what it needed to do on the plant. And based on that argument earlier that it was the amount of carbon on a plant that was driving how many of these microbes were present, why not we naively thought that it was simply auxin was being produced by the bacterium to somehow cause more nutrients to come to the surface. But it seemed like it's probably a little more complicated than that, because we had other biological sensors that we developed. In this case, we made a biological sensor for sucrose, where we took a sucrose responsive promoter, and in this case, drive a promoterless isonucleation gene, and then ask how many isonuclei could be made in the presence of the sucrose. The thinking being then that we compared a normal plant in the purple with a plant where we had extra auxin being applied, that there ought to be more sucrose available because the auxin was going to somehow make nutrients like sucrose more available. But it was the opposite. We did it over and over. We always got the wrong answer. In this case, we saw less sucrose present on the plant in the presence of the auxin compared to the quote normal plant. So less sucrose was available in the presence of auxin, but the opposite was true for fructose. We already described the fructose reporter. In this case, we see in the green compared to the blue, the presence of auxin actually made fructose much more available. Lower sucrose, more fructose. Okay, well, what is this trying to tell us? But then we turn to think about, well, again, what is life going to be like on this leaf? Because go back to those 10 micrograms of sugar that we talked about on a whole leaf. And on a leaf, it gets wet from rain or dew. You dissolve 10 micrograms of sugar in that solution. It's going to have to be really dilute. It appears then that, in fact, these microbes are really good at eating very dilute carbon sources. But more importantly, they have active uptake mechanisms for these different sugars. In this case, fructose is taken up by a different mechanism than that of sucrose. And more importantly, those uptake mechanisms for the monosaccharides like fructose are much more efficient than the uptake mechanisms of sucrose. So we tried growing the bacteria at these very, very low concentrations we would expect on a leaf. And the units are important. So we're looking at a fraction of a milligram per liter of carbon, which is what we think is the concentration that they actually see. Nothing like the 10,000 times more that we would typically grow them in the laboratory. And more importantly, what you can see is that they could start growing at like 0.02 milligrams per liter. Really, really low concentrations. And it took about 50 or 100 times more concentrations of sucrose for them to also start responding to the sucrose to grow. So it looks like they really can't grow at the expense of sucrose on the plant because it's never a high enough concentration. And what they're actually doing is converting in some way or another sucrose to the monosaccharides, fructose and glucose. So it appears then that what is happening is that this auxin that the bacteria are making are acting on the plant to induce invertases, the enzymes in the plant that convert to sucrose, which remember is the dominant carbon source that's available in or on the plant, to the glucose and the fructose that the bacteria can actually eat at those low concentrations in the plant that the plant is providing. Now, one last trait I want to mention then is quickly these biosurfactants. And When you'll see dewdrops on a plant or raindrops on a plant, the leaves of most plants are relatively hydrophobic. They've got a hydrophobic cuticle. The cuticle is there to prevent the nutrients from escaping and also preventing water from going out of the plant when it's dry. But they're usually pretty waxy surfaces, and it's not uncommon to see, drops of water on the surface. Much of the surface would actually be not colonizable because there's not going to be any water present in most of the leaves. So we thought naively, yes, many bacteria have the ability to produce detergent-like molecules, kind of amphipathic molecules with a hydrophilic end and these long acyl chains, which act like detergents. And Pseudomonas syringae is one such biomimetic syringifactant that's known as. We thought, well, it's going to allow the spreading of water on the leaf to allow it to be wetted better. But it turns out that the syringifactant, the detergent, has two other properties that seem to make a lot more importance to the bacterium on the plant. And one is to change the nature of the cuticle on which the bacterium is sitting. So we could test that by peeling off tiny bits of the cuticle away from just a very thin layer on the surface of the plant. We peel it off, put it on top of a chamber where we had some tritiated water, and watch the tritium diffuse across that cuticle into an upper chamber. And that's what we're seeing on this graph, basically the accumulation of tritium. So an intact cuticle allows a slow diffusion of tritium, but then when we applied some syringifactant, you see the rate at which the tritium is now moving across that cuticle went up dramatically. So it seems like it's naively dissolving that cuticle. It's somehow modifying the nature of the cuticle to make it more porous to polar molecules like water or maybe some of the polar sugars and so on. So that is one of the traits that many bacteria have the ability to produce these detergent-like molecules. And the other trait that I wasn't really aware of until we started working with syringifactant is that many of these type of molecules are very hygroscopic. They bind water. In this case, the syringifactant made by Pseudomonas syringae can bind 2 to 5 times its mass in water. So again, bacteria are by nature aquatic organisms. Here we're having them grow on a frequently dry leaf. So having the ability to somehow hold on to water might be quite important. So we wanted to know, was the presence of this detergent-like molecule that could bind water making the bacteria more hydrated or less water stressed? And we could again develop and use biosensors to have the bacteria tell us how water stressed was the bacterium. So in this case, we used a promoter for proline biosynthesis. Proline is one of these common compatible solutes. Proline makes water more accessible to the inside of cells. So we're going to link that again to a GFP reporter, and as the cells are going to see water stress, they're going to try to make proline to deal with the water stress, and they're going to turn green. The greener they are, the more water stressed they will be. And so we put that bioreporter gene into Pseudomonas and compared how green the wild type strain became compared to a mutant that we made that was blocked in the production of the biosurfactant. What we're looking at is the distribution of the greenness of a large number of cells. Each of these little dots is how green a particular cell was. So here's a fraction of cells as a function of how green they became. Basically, we're seeing that the wild type cells never got particularly green, not anywhere near as green as did our syringifactant mutant, suggesting that they did not have the ability to hold on to the water. And when we put them on a plant and exposed it to a typical environment, they were more water stressed. So it did seem like our simple model was probably correct, that the bacteria making the syringifactant, that you see these little balls and chains here, the amphipathic molecules that surround the bacteria, were now binding water, and the cells were essentially sitting in little puddles of water. Maybe it's a little simplistic, maybe it's a little film, but basically they were making the bacteria much less exposed to the dry environment that they otherwise could tolerate. And it was quite true, though, that compared to the survival in the field, the wild type does much, much better in a field condition where we put these recombinant strains compared to the mutant, which is on the bottom. So it looks like then it was very much a local environment where the cells were making themselves more wet. Now I want to finish up talking a little bit about the bigger question about how do things get onto the plant in the first place? And remember, we tried to convince you that certainly young tissues like flowers or young leaves as they first emerge into the world are largely uncolonized. So where is the inoculum coming from to grow on those plants? And if you could see in this apple orchard picture, it would seem likely that there would be a lot more bacteria available on the surface of these grasses than would ever have been present on these young flowers. Flowers are virtually sterile. In this case, in California, the grasses have been growing all winter. They have 1 million or more cells on a given blade of grass. So there's a huge number of bacteria there compared to the flowers. And could there be movement of those microbes up onto the leaves? We're thinking it's airborne, but it might also be contributed by insects that might fly between the two. But studying first the airborne aspect of this, we did a simple experiment. Berkeley is in the San Francisco Bay Area. So we face the Pacific Ocean. We're on the east side of the Pacific Ocean. The winds almost always blow from the west. So the winds are coming off the ocean. They haven't seen any plant material since it left Asia, 3,000 miles away. So we could take pristine air as it was coming off the ocean, and we took vacuum pumps down to the edge of the water, and we could sample a cubic meter of air onto filters. And we also had other vacuum pumps that would bring another sample simultaneously, in this case, about 30 meters inland. So there were grasses and things that went right down to the edge of the water. So we would then quantify how many microbes were in that cubic meter compared to that cubic meter, the upwind and downwind air, if you will. And it was much more dramatic than I would have expected. And when you compared the numbers of microbes that you saw above, the upwind air had about, the downwind air had about 6 times more bacteria in it. Even after the air had flowed only, about 3 times the size of this room, it had already been populated by airborne bacteria, presumably from the plants. And we could do analysis of the communities that we had captured on our filters by DNA analysis of the signature genes. And, here's a list of the kind of taxa that we found most abundant in those air samples. And we're going to compare the upwind air, the communities on the plants over which the air had flown, and then the downwind air, that 30 meters inland. And just look at a couple of the more typical bacteria that we see on plants, like Pantoia, very common bacteria on many plants, was very abundant on our plants as well. And then it was also quite abundant in the downwind air. It basically was not present in the upwind air. And you could pick out some of your other favorite bacteria. But it basically showed us that the air, even after a relatively short distance, had become much more plant-like in the communities that it was harboring. So that led us then to think then that the communities that are on the plants, if it's being inoculated by airborne bacteria, might be strongly dependent on the communities of plants nearby. I won't show you the data that seem to suggest that soil is not a particularly good habitat to get the bacteria up on the plants. It's probably other plants. There are unique traits that plant-associated bacteria have. And so again, if you have a community, if you got a context like this where you got different kind of plant species around, it's probably going to be a very different process depending on who your neighbors are, because they're going to be the sources of the airborne inoculum. Now I want to finish up talking about the way we're, again, messing with the world in agriculture, because we're growing corn and whatever in these large fields where there aren't any other green plants in the vicinity. But the first question then is, how context dependent is the communities on the plants? If as a given plant species, are you the driver? Is selection happening because of the characteristics of you as a plant host Or are you going to be simply sampling from many other potential colonizers? Are there a variety of microbes that you could harbor, and you're kind of at the whims of who your neighbors are supplying that inoculum? So my brother is a farmer in Oregon, in the northwest part of the United States. And he raises in large fields 3 major crops. One is fescue. It's a common grass for pastures and lawns and things. He raises crimson clover, a really pretty crop, and wheat. And so what I did, I used fescue as like a sentinel plant. We grew fescue in the greenhouse so that it had many other bacteria on it, and we transplanted them out into the middle of his field. So here you can see some of the easiest to see in this picture where the red clover has got some fescue plants that I put in the middle of it. We came back a couple, 3 weeks later, and we harvested the fescue plant to see what was the communities. And if it was driven by selection from the plant, we would have expected that the communities on the fescue would have been very similar no matter what setting we had put them in. But in fact, it was very different. And I don't remember which is which. The yellow was the wheat and the red was clover or whatever. You'll see in this principal component analysis plot that the communities were very different. These fields aren't that big. There was in about a kilometer or one or so. But it was their local neighbor that was seen to be driving what the communities were. There was a substitutable community on the plant, and it was driven by the nature of the local plant community. So this had a lot of implications in terms of agriculture. I'm interested in a lot of diseases and frost and agriculturally relevant microbiology, if you would. And frost especially is a big issue in California. We raise a lot of citrus, navel oranges and tangerines and other things. They're subject to freezing in the wintertime. But the orange groves are grown in different kind of contexts. In this case, you see a grove that was grown next to this field of grass. Many of the orchards are large, and they've grown only in large blocks where their neighbors all were other citrus trees. And they usually remove all the vegetation in these orchards with herbicides to prevent competition for water and things. So here we would have had a setting where we knew that there was probably 1,000 times, maybe 100,000 times more bacteria per unit area of those grasses than ever would have been found on the citrus trees, especially following the summer. Our summers in California, there's not a drop of rain for about 5 months. It gets about 43 degrees Celsius. Terrible place to grow if you're an epiphyte. But then winter comes and it gets cool and rain. And the communities we then think happen from these communities coming off of these other plants that were around by. So what we looked at then was the communities on these citrus trees very different in this setting compared to this lower setting? And so again, we looked at transects where we had obvious sources of inoculum, like in this case, this is the edge of the citrus grove, and there were other grasses and other things here. And we were going to look different distances into the grove to see whether the communities on the edge of the grove, where it's very close to the grasses, and then move into the large groves. So here we collected not only airborne bacteria, not with vacuum pump, but by just watching them deposit onto petri dishes for an hour. And you could see that there was a very dramatic difference in the microbiology of the air, if you would. In this case, we're looking at the deposition. These are how many culturable bacteria we deposited in an hour on a petri dish. And we had this on several different orchards with different settings. The green ones were where we had surrounded these grasses nearby. And the red one is where it was surrounded only with other citrus, where there wasn't a real obvious source, if you would. And you'll see two things. The red ones are almost all much lower in the rates of deposition of the bacteria than was the ones sort of where there were grasses nearby. And you see the green one especially, see it slopes downward. So here is right on the edge of the grove next to the grass. And as you move into the grove away from the grass, you see that the rate of deposition decreased and decreased and eventually got to this low level that you saw when we were in an area where there wasn't an obvious source. That was associated then not only with the rates of deposition, but the actual numbers that we saw on the trees. So the green ones again are the communities, this is log cells per gram. Again, we see the green ones show a slope downward. There's about 10 times fewer bacteria per unit area on the interior of the grove than on the edge. And again, the green ones were, for the most part, all higher than any of the red ones, where there wasn't an obvious source of inoculum nearby. And fortunate for us and unfortunate for the growers who were collaborating with us, we had freezing events that happened in these groves. We had already measured the supercooling temperature. We could put these leaves in tubes of water and cool them to see when they would start to freeze. That's what we call the supercooling temperature. And you could see that those leaves that were collected from trees that were adjacent to citrus had a much higher ability to supercool compared to those on the bottom one where they were just adjacent to grass. And again, we see this strong gradient. There was much less supercooling near the grasses and much more supercooling as you moved into the grove. So 25 trees in was about 100 meters or so. And the frost that we eventually saw showed the same pattern. We actually had damage to the fruit that were hanging on the tree, more damage near the edge of the grove where there was the bacteria present, and probably the ones that didn't have the grasses present, there was much less freezing events. Maybe a little more understandable would be some similar work that we've done with grapes. Grapes are a big crop in California, obviously. And the highest value grapes are grown in the northern part of the state where freezing is quite common in the springtime. And based on what we had studied with citrus and the airborne bacteria at the beach, we were very worried about what the effect of these agricultural practices like the growing of cover crops between the rows. So this area where the high value grapes are grown is kind of hilly. Erosion is a problem. So they often would want to have vegetation there during the winter to kind of stabilize the soil and improve soil health and so on. But we thought it would be a major risk to the grapes if that was in fact providing a source of inoculum that would move up onto the newly developing grape shoots. So we set up some very large plots, replicated blocks. Each block was about 3 or 4 hectares in size because we needed to get far enough away from the other sources of inoculum to show that. So here's where 2 of these replicated blocks came together. Here where we had removed the vegetation early in the winter with herbicides, and another where we had allowed the vegetation to grow. And then we collected the tissues from the grapevines and looked at, in this case, supercooling capabilities. And the hot colors, the yellow and the red, are where we had removed the vegetation, and the color, the blue and the gray, is where the vegetation had been allowed to be there. And the y-axis is showing, again, the supercooling point. The more supercooling reflects the lower numbers of ice-nucleating bacteria. And you can see quite a substantial effect, about 2 to 3 degrees Celsius colder before those plants were likely to have frozen had frosts occurred because of the lack of the inoculum of, in this case, ice-nucleating bacteria that presumably were coming from the grass. So just want to finish up and talking about agriculture. I came from an agricultural background. I think our fundamental work that I've been talking about has important implications in terms of growing plants correctly. And again, this is the way most of the developed world grows plants anymore, in huge fields without any weeds. And the real question is, where is the inoculum going to come from on these large fields? Because remember, we were talking about, the grass is right next to the grape. But in a large field, if you're in the middle, where is the inoculum going to come from? So we did some experiments to see how much of an edge effect is there? Will you see a different pattern of colonization of the plants, in this case, of little corn plants next to some stuff, okay? And so we did a large experiment. We're right in the middle of it. I'll show you the preliminary data. This is a field in Minnesota. This is my postdoc, Kyle. And we were fortunate, we had these gigantic fields. So you're only seeing just one edge of this huge field that was about 100 hectares in size. But they bordered on nice green trees and grasses and things on the edges. And what we were doing then is to collect these developing corn leaves near the edge of the field and then moving further and further away from the vegetation to see what we would find. So this so far is what I can show you. Let's just look at the corn as a good example. So we were looking only when the plants were relatively young because we thought that would be the most important time to look at the colonization process before everything became green and they shared inoculum among themselves. And this is now based on DNA analysis, the total numbers of bacteria that were found on our edge plants, those trees and everything we saw. Indeed, they do have like 10 to 50 times more bacteria on them early in the spring. But more importantly, look at the gradient as we move into the field. So we got about 100 meters into the field. You could see that the total numbers of bacteria on these young corn plants after a week or so after they emerge from the soil are about 10 to 20 times lower than they were near the edge. And we've also done community analysis. We find out who's on those plants in addition to how many were on the plants. And we see a very different pattern that the bacteria on the edge of the field look a lot like the surrounding plants, and the ones on the inside look substantially different. So it would seem then that there really is a fairly large kind of biogeographical feature to the colonization, especially early in the development of these tissues. And I would just summarize everything we talked about today, is that the colonization of plants seem to involve very different processes that happen at different scales. So early on, we talked about all these genes and surfactants and what have you. We think that's all happening at very, very small scale, probably within micrometers. These bacteria are tiny relative to the large leaves that they're sitting on, and that they can only influence the local areas of the leaf. But they do need to alter the leaf surface to make water more available or causing sucrose to be converted into fructose and so on. We think that's all happening at very small scale, but at big scales, this whole immigration process, which is important in getting organisms on the plant, is also really important. So I think we have to kind of think of it as a very complex process that's actually going to be happening. My final scale point then is that, again, humans are messing with the world. It's pretty clear. Climate change is happening. We talk about carbon dioxide, but think back to the, climate brightening of the clouds and everything. But agricultural especially, we think that there are problems in the way we are growing plants that might make them much more susceptible to diseases by eliminating what would be a normal community that would happen on these plants. Especially if we have pathogens that might emerge from, say, the debris from a previous crop here onto a young developing plant. It might be arriving on a plant that's under colonized and is going to be more susceptible to invasion by the pathogen. So how can we avoid that? If we're not going to grow them in huge fields, maybe we should inoculate those plants with beneficial organisms. And if you follow the agricultural microbiology field, this is really, really big these days. All the corn and soybeans in the United States anymore are inoculated with different microbes, usually to stimulate root growth and what have you, but we think might also include microbes that might also restore more of a natural community on the leaf surface. I'll just stop there, and while I'm having fun in New Zealand and all, these are the people working on the hard part in the lab. Despina did a lot of the aerobiology. Adrian worked on our biosurfactants. And Monica did our biosurfactant work, gene expression. So had a great crew to work with. Thank you much.
Great, we have time for questions. Teaching's finished, so I don't think anybody. So the primary limitation is actually when Steve is taken off to Hobbiton. How much time do we have?
I'm cool.
You go. Okay, great. Any questions in the room or on Zoom? Margaret first. Sorry. All right.
Can you go over the superciliating? So I'm not sure I understood it, but with the hypothatic bacteria and citrus orchard, the ones that had that were closest to the grass, do they have the citrus trees have more ice nucleating bacteria on them than the ones closer, the ones in the grove?
I'm having a hard time hearing. Can somebody summarize for me?
With that ones, okay. Hello, hi. I just had a little bit of trouble understanding the supercooling effect and the ice nucleating bacteria. If you could just clarify. So with the citrus orchard, the citrus trees closest to the grasses had more ice nucleating bacteria on them. So what effect did that have on frost damage? Was that a beneficial effect or a detrimental effect? I didn't quite understand that.
Most of the agricultural plants that we grow are sensitive to freezing. So in this case, the grasses would be considered a risk factor for, in this case, oranges or flowers, and you name it. So that there's a few plants for which ice nucleation is beneficial, but it's kind of a unique thing. Most of our plants are very intolerant of ice formation, and those bacteria and the nucleators that would move are going to be real much of a risk.
Okay, so in the citrus orchard and the grape example or studies you showed us, it was actually a detrimental effect to have a cover crop or other species growing around the desirable fruit.
Same thing, the cover crop being very beneficial for soil water. There's a trade-off in terms of increased frost risk and all.
Okay.
So I thought you were going to say, oh, if I was a grape grower, I worry about my soil health, right? And the cover crops are going to be very important for that, and erosion and all those other things, but there may be a major risk for the frost damage. I didn't talk about it, but we've been looking to see, are there plant species that could be poor colonizers, poor hosts for the ice nucleating bacteria that you might intentionally grow in between your rows? And the answer seemed to be kind of yes. There are some plants vary in how many of these nucleators they'll support. And so there are certain legumes and things we found that might be a good compromise in terms of allowing the beneficial growth of your cover crops, but without the risk of the immigration of the nucleators.
Okay, thank you. That's really interesting.
Really enjoyed that. Thank you very much. I have a question about bacterial. Whether you looked at bacterial distributions within the leaf, whether there were regions within the leaf that had these aggregates of bacteria?
That have
Aggregates of bacteria, like on the surface.
one of my students did a lot of work on that kind of local geography on the leaf. There's two big places you'd most likely find them. The most common place is at the base of glandular trichomes. So certain plants have these little hairs, and at the base of the hairs, they have the ability to secrete chemicals onto the leaf, often for defense against insects and what have you. But they seem to exude nutrients as part of that. And so those are the most likely places to find them. The other places along veins, and that might be partly because of the escape of nutrients from the phloem that's exposed most prominently at the veins, but partly a physical phenomenon because you get what's called the coffee ring effect that also occurs on plants, where as the water evaporates from the leaf, it evaporates last along the veins and things, and it kind of draws any bacteria that might be suspended in the liquid to those last evaporating places. So you get this coffee ring effect with bacteria physically moved around by the water as it evaporates.
The reason I ask about inside the leaf is that there's a current debate about the relative humidity, if you like, the saturation point within leaves inside. And there's some models that suggest there's recondensation of water. So there's wet patches or wet locations within the leaf. And I was just wondering whether bacteria are found there or not.
The numbers of cells inside of a plant vary quite a bit, but they're almost always much less abundant than they're on the surface. And some bacteria passively get into the leaf and seem to just accumulate there, but in relatively low numbers. If they're pathogens, then they can actively change the nature of the leaf and they can grow to huge numbers, but assuming we're not talking about a pathogen, the numbers that are inside of the leaf are 1% or something, what we might see on the surface. Yeah. And whether those are in particular places where there are wet patches or what have you, not much has been done on that.
So I have one question of my own. So in doing this work, have you identified morphological or physiological traits of plants that are predictive of their ability to harbor ice nucleating bacteria?
The number, yeah, particular features are the most predictive. The waxier the plant, the harder, the waxier, smooth surfaces like a citrus leaf. Or things like peas versus beans. Peas have kind of a waxy leaf, you know, and when you see the wet, water makes little drops on top of a pea leaf, whereas other plants it makes kind of a film. It's that the wettedness of the leaf surface, how thick the waxy cuticle is, and the structure of waxes. Waxes are make little shapes like, you look at the magnification of a leaf surface, it's kind of a looks scaly like a snake or something. The wax crystals make a rough versus a smooth surface, and the smoother those waxes lay down on the surface, the harder it is for water to escape.
Great, thank you. Any more questions? Oh, sorry, two more.
I'm sure you're familiar with the movement to establish like prairie patches around, you know, areas of farmland that aren't in use. And how do you think it would be best to balance the desire to restore the function of the prairie and potentially the cloud formation with the potential damage to the crops nearby with more ice nucleating bacteria?
I've been interested in that. In entomology, there's been a lot of interest in these strips that, like in England, not much here, but in England especially, you almost always see like hedgerows and things around the field. And those have always been kind of all that is really a plus because they are good reservoirs for beneficial insects and all. We think that might also be a source of these bacteria, but their big issue that we're trying to get out of some of these studies is what would be the size needed to be a good source. So my worry is that if we have to dedicate a large fraction of the area to something that's unproductive, it'd never be practical to have enough of these dedicated sources of inoculum, if you will. That's why these gradients that we're trying to get at in the field, or what we're getting at is how far of an effect can you expect from these sources of immigrant inoculum? And if it's a very short source, then you can imagine it will take a lot of these preparations to make it work. So I don't know the answer, but I'm worried that it will be too many.
Thanks.
Oh, yeah.
Thank you. Yeah, thanks, Steve. Great presentation. Really enjoyed it. You covered really a lot of ground in that presentation. And when I try to put things together, the fact that you demonstrated that the ability of a pathogen to cause disease is partly influenced by its neighbors. And I guess the reciprocal, it's true that the ability of a biological control agent to prevent disease is also controlled by its neighbor, and that the bacteria that end up on the plant are completely dependent of where that plant grows. It seems to me it brings into question how much you can extrapolate experiments you do in one setting to another. In particular, I guess the extreme example would be, can we even think of extrapolating what you do in a glass house to a field? So
yeah. Well, first of all, the glass house, we grow our plants in the greenhouse, most of it in the first place. We know they're underdeveloped in so not our control always. But as you were talking, I was thinking a lot of what we're trying to do practically is the kind of things that you've done and I've done as well in terms of inoculating plants. If we can't get enough sources of natural dispersal into our plants and flowers to get the protection, can we artificially inoculate them to achieve the same goal? And I think in the ultimately, that's kind of what I was finishing up with these probiotics that a lot of the big companies are now selling microbes for specific inoculation on the plants. And that may be our only practical solution, is to artificially create colonized plants. And especially if we could figure out how to make it work well from simply seed inoculations for an annual plant, because we can't think about spraying the vast acreages of corn or something with these microbes, but they're already being inoculated with inoculation onto seeds. If there were particular strains that could grow well on the seedling as it developed from the seed, that might achieve some level of the normal community. But that's going to be a lot of work too.
Any more questions? Well, if not, I'd like to thank Steve for having this wonderful seminar. One last round of applause. Okay.